Thank you, Jack, for the kind introduction. Welcome also from my side, and thank you all for joining us. Today's presentation will take about 45 minutes, and as Jack already mentioned, we will take care of your questions right after it. So let's start now with our webinar by having a look at the contents. First, we will briefly touch on the types of contaminants, because if you fight something in the lab, you would want to know what you are fighting against. We will also speak about contamination sources and some easy ways to prevent contamination. At the end, we will go through just a couple of tips for aseptic handling. Contamination is an everyday challenge in a cell culture lab. All labs are prone to contamination, but the frequency varies. All labs have contaminations from time to time, but understandable, nobody likes to talk about it. I think we can also agree that contaminations cost time and money. Undetected or ignored contamination actually endangers the reliability and reproducibility of our results. So what do we do against it? In the majority of cell culture labs, 70% ethanol is the #1 substance for disinfecting surfaces, equipment, etc. The good thing about ethanol is that it is very effective against bacteria, yeast, and some viruses. It is the only disinfectant which evaporates completely residue-free. Therefore, it is well suited for routine rapid disinfection, for example, of the work surface in the biosafety cabinet and equipment that is used in the hood. If possible, you should prefer wipe disinfection over spray disinfection, not only to prevent aerosols, which are harmful when you inhale them, but also to enable a complete wetting of the surface for proper disinfection effect. When using ethanol, you should keep in mind that it's not effective against all kinds of contaminants. Bacterial spores, for example, can survive alcoholic. Although ethanol has a lethal effect on fungi, it takes a few minutes until the fungi are killed. Due to the fact that ethanol evaporates within seconds, it is not really effective against fungal contamination. So if you are facing ongoing problems with fungal in your cell culture lab, it may become necessary to implement a cleaning routine with a disinfectant which has a broader spectrum. The second thing many researchers worldwide use to fight contaminations are antibiotics. This used to be a good tool, but it is becoming more and more a poor tool because there is limited protection due to the increasing resistance of bacteria. The most commonly used antibiotics in cell culture are penicillin and streptomycin. Their non-cytotoxic effect was first described in the 1940s, and they are still present in most cell culture labs. As you can imagine, in over 70 years, the continuous usage of these substances supported the increase of resistant bacteria tremendously. So does it help to simply switch to other antibiotics that are not so common than penicillin and streptomycin? Well, it may be a short-term solution in some cases, but in the end you are facing the same issue with resistant bacteria. A more general aspect of the routine use of antibiotics in cell culture is that it gets more difficult to detect microbial contamination. Without antibiotics, most of the introduced contaminants would soon be visible under the microscope, or even with the naked eye, as color changes and turbidity of the culture medium occur. The prophylactic routine use of antibiotics in cell culture not only masks poor aseptic techniques, it can also leave low level of partially resistant contaminants undetected for longer time periods and therefore creating a greater risk for the whole lab. Besides the effect of antibiotics on bacteria, what most people ignore is that it is known that antibiotics can have side effects on the metabolism of cells. Here are two recent studies describing effects on different cell types. Whereas the usage of standardized culture media and their supplements is widely established and accepted, the prophylactic usage of antibiotics varies greatly. Different researchers use different types and concentration of antibiotics for the same cell type. Some researchers don't add any antibiotics to their cell culture. So in most cases, the data that are published for a certain cell type were generated under non-standardized conditions. This, of course, can be problematic when it comes to reproducibility of experimental data. So taken together, antibiotics are not really good in protecting the cells from bacterial contaminations, and they are not really good for the cells themselves. This is why we would like to show with this webinar that there are simple and efficient ways that offer huge potential to protect your cell culture from contamination.